Trees build a fortress of lignin that resists everything. Yet fungi break it down like a secret code. How can microscopic enzymes dismantle such a tough polymer? The answer lies in tiny radicals that hijack the bonds. Watch how they do it, and why we still can't replicate it. Now that the mystery is on display, let's peel back the layers of lignin itself. Lignin is a complex aromatic polymer woven into every woody cell wall, giving trees their strength and resistance to decay. Fungi have evolved specialized oxidoreductases lignin peroxidase, manganese peroxidase, and laccase to attack it. These enzymes generate phenoxy radicals that break the toughest bonds. Lignin is a complex aromatic polymer that fills the spaces between cellulose and hemicellulose in plant cell walls. Its structure consists of phenylpropanoid units linked by ether bonds, such as beta-4 linkages. These linkages give lignin its rigidity and resistance to chemical attack. In forests, lignin accounts for roughly 30% of the dry mass of woody biomass worldwide. Because it is so abundant, lignin represents a major reservoir of carbon in terrestrial ecosystems. When trees die, their lignified tissues become the substrate for specialized fungi that can break down this polymer. Fungal enzymes such as laccases and peroxidases oxidatively cleave the ether bonds, releasing phenolic fragments. The degradation products are then mineralized or incorporated into soil organic matter. This process is essential for returning carbon to the atmosphere as CO2 and for maintaining soil fertility. Understanding lignin chemistry helps us predict how forest carbon stocks respond to climate change. Pause 0.8 White rot fungi initiate decomposition by secreting oxidative enzymes that attack lignin. They produce laccases, manganese peroxidase, and versatile peroxidase to break down aromatic rings. This enzymatic assault creates phenolic fragments that diffuse into the cell wall matrix. Brown rot fungi, in contrast, rely on a chelator-mediated Fenton reaction to generate hydroxyl radicals. These radicals cleave cellulose chains but leave lignin largely intact. The result is a porous wood structure rich in hemicellulose and lignin residues. White rot enzymes are secreted into the extracellular space, allowing diffusion throughout the substrate. Brown rot hyphae secrete fewer enzymes. Instead, they concentrate on rapid depolymerization of cellulose. Consequently, white rot fungi recycle carbon more completely than brown rot species. Understanding these mechanisms informs forest management and biofuel production strategies. Pause 0.8 Lignin peroxidase initiates degradation by oxidizing a phenolic group on the lignin monomer. The enzyme uses hydrogen peroxide as an electron acceptor, forming a high-valent iron intermediate. This intermediate abstracts a hydrogen atom from the substrate, creating a radical that propagates chain scission. Manganese peroxidase follows a similar pathway, but first oxidizes Mn-Roman 2 to Mn-Roman 3. Mn-Roman 3 is then released into the matrix and chelates phenolic sites on lignin, facilitating oxidation. Laccases are copper-containing enzymes that transfer electrons directly from a substrate to oxygen. They generate water as the sole byproduct, making them attractive for biotechnological applications. All three oxidoreductases share a common strategy. Electron removal creates radicals that break aromatic bonds. The resulting smaller phenolic fragments are more soluble and can be further metabolized by microbes. Thus, lignin peroxidase, manganese peroxidase, and laccase act in concert to recycle carbon in forest soils. Pause 0.8 enzymes first bind to the lignin polymer. They position a catalytic iron center nearby. A hydrogen atom is abstracted from an aromatic ring. This creates a phenoxy radical on the lignin. The radical destabilizes adjacent CO bonds. Bond cleavage follows, breaking the polymer chain. Subsequent rearrangements form smaller aromatic fragments. These fragments can be further oxidized by other enzymes. The process releases energy that fuels microbial metabolism. Thus, lignin is gradually mineralized into CO2 and water. The radicals also facilitate cross-link cleavage. Overall, this mechanism drives carbon cycling. Pause 0.8's lignin depolymerization begins with oxidative cleavage of aromatic rings. Enzymes such as laccases generate radicals that attack the CC bonds. Radical intermediates rearrange, forming phenolic monomers like vanillin. Side chain oxidation follows, converting methoxy groups to aldehydes. Syringaldehyde emerges when two methoxy groups are retained. Further oxidation yields protocatechate, a key intermediate for mineralization. These soluble phenolics diffuse into the soil microbial community. Microbes then catabolize them via beta-catodipate pathways. The reaction network diagram links each monomeric unit to its products. Understanding these steps clarifies carbon cycling in forest ecosystems. Pause 0.8's lignin degradation releases aromatic compounds into the soil. These compounds serve as carbon sources for microbial communities. Fungal hyphae transport degraded lignin products deeper into the litter layer. The movement of these molecules fuels bacterial respiration and CO2 evolution. Simultaneously, nitrogen-bound in lignin is mineralized to ammonium. Ammonium becomes available for plant uptake or further microbial use. Thus, fungal activity links wood decay to soil carbon turnover. It also regulates the balance between carbon sequestration and release. In mature forests, this process sustains long-term nutrient cycling. Understanding these mechanisms helps predict ecosystem responses to climate change. Pause 0.8's engineered fungi can produce more peroxidase enzymes. These enzymes break down lignin faster than wild types. Higher activity means more cellulose becomes available for fermentation. In a bioreactor, the engineered strain is grown on wood chips. The reactor's temperature and pH are tightly controlled to maximize enzyme output. Lignin fragments released into the liquid phase can be collected for biofuel precursors. Simultaneously, toxic phenolic compounds from industrial waste are degraded by the same enzymes. This dual action demonstrates how lignin degrading pathways can aid bioremediation. Future work will focus on genome editing to further enhance peroxidase stability. Such advances could reduce costs for both biofuel production and pollutant cleanup. Pause 0.8's lignin's breakdown by fungal enzymes turns resilient wood into soluble phenolics that fuel soil carbon cycling.